So if you were here at CGSI last year, Adam Park, a master's student, actually gave one of the 10-minute talks on this subject. So this is going to be a bit longer, a bit more detailed version of that. So plan for today, I'm going to motivate the problem, talk about the actual Prokrustean graph, this new data structure that has to do with k-mers, and then look at a couple of applications, and then conclude and point you to the code. In the back, can you guys hear me? Because I have like speakers right here. Okay, awesome. Cool. Okay, so to motivate the problem, thankfully Ryan motivated this quite nicely in the opening talk, talking about the history of k-mers in bioinformatics. Basically, we got long strings, k-mer, length k substring of that long string used all over the place in bioinformatics. Used in assembly, in classification, in similarity searches, in alignment, et cetera, et cetera, et cetera. So they're rather ubiquitous in their applications. And there are a few different reasons why this is. So them being fixed length makes it quite nice for us computer scientists to deal with. Allows for easy, fast matching of these things. Exact matching is very nice. It's very easy to talk about locations of mismatches because everything is fixed length. And it's easy to parallelize over all these things. Many applications, for good or for bad, treat k-mers as independent from each other and can parallelize their tasks, as well as you can do a lot of sketching stuff with them. But you have to pick a k-size to use k-mers in one of these applications. And at least from my perspective, the question of what k to pick really is a rather large elephant in the computational biology room. Because if we look at the range of different k-mer sizes that are used from 0 up to over 150, which is basically an entire read if you're using short read Illumina kind of technology, different bioinformatics techniques span this entire spectrum. So k-mer 2vec uses 2-mers kind of thing. And then as you increase, maybe this tool uses around 20, and then Spades comes in 3 different flavors that uses k-size all the way up to 100 plus. And so with this huge range of k-sizes, qualitatively and quantitatively, things change pretty significantly. And there is a little bit of, I don't know, folklore that you can abstract from this situation in that small k-mer sizes are usually used for like very crude, large-scale kinds of signals, things that are kind of obvious, like, I don't know, classifying things at the phylum level sort of thing. Whereas a little bit bigger, maybe around the 20-ish range, this seems to be what most people use for doing general similarity measures and alignment kinds of stuff. If you really want to tell the difference between like 2 different strains of bacteria, then you might use an even bigger k-mer size like this. And then these really large k-mer sizes are usually reserved for long-read genome assembly sorts of things. But there's no good theoretical reason for why these different regimes for the different k-mer sizes are. This is basically folklore. One of the reasons why this is it's really hard to formalize what is the best k to use. Like we heard a question in the first talk saying, oh, an optimal k. Well, what does it mean to have an optimal k? It's really hard to do this because optimal k is tangled up in your application. So your bioinformatics pipeline deals with some sort of k-mer-based object that is dependent on the k-mer size and the sequences. And then you have a whole bunch of biological adjustments and then engineering thrown in and stuff like this. So it's really hard to talk about optimal k for an entire computational biology pipeline. So instead, what we're going to do today is sort of zoom in and focus just on these k-mer-based objects. How do these things change as a function of k size? So what are these k-mer-based objects that I'm talking about? Well, for one thing, counts. So k-mer frequency, histogram sorts of things, occurrence, presence, absence kinds of things. I want to talk about metrics thereof, of counts or presence, absence. I also want to talk about extensions. So for example, a De Bruijn graph, if you're familiar with that, you're talking about, oh, can I do a right extension off of one of my k-mers? As well as occurrences, particularly near heads and tails of sequences, used in things like overlap graphs. So let's look at a few examples of these different kinds of k-mer-based objects, like counts. You have a certain sequence, and you look at the k-mer count. Well, interestingly, how the number of distinct k-mers behaves as a function of k-mer size is non-monotonic. right? So notice here we're increasing from k-mer size of 2, 3, 4, 5. The counts go up, stay the same, and then start decreasing. For a single finite length sequence, there's some nice theory back from the 2000s that describes this sort of behavior. To actually compute this in practice, you could do this pretty easily with something like a suffix tree. So you take a suffix tree, and then you look at the different levels of the suffix tree, and that's going to give you the number of distinct k-mers for different k-mer sizes. Okay, cool. We have theory, we have a data structure. And people do this in practice. So even just, I guess, last year, the number of distinct k-mers as a function of k-mer size was used in genomic sequence similarity comparisons. Okay, so single counts, that's pretty good. What if we start moving to metrics? So metrics, now we have two sequences. So here I've concatenated sequence 1 and sequence 2 together. We might talk about some sort of metric on these two, like the Jacquard index. K-mers in one, k-mers in the other, k-mers in both or just one of them, and then the weighted version of this. This is slightly more annoying than single k-mer counts, but still doable. So here again, the behavior is non-trivial. As you increase the k-mer size, what happens to the number of shared k-mers? And again, we have a data structure that you can use this with. So for example, with the suffix tree here, now you also have to keep around an array. An array tells you whether these different nodes or edges in the suffix tree appear in only one of the sequences or both of the sequences and what their counts are. So you can do it, it's just a little bit annoying. And then we move on to de Bruijn graphs. So extensions. So recall what a de Bruijn graph is. We have our big old genome. Let's say that you sequence it with a bunch of reads, and you take the k-mers from all of those reads. The de Bruijn graph has nodes which are k-mers, and the edges between them represent that the tail overlaps with the head of all but one character in that k-mer. So this would be an example of a de Bruijn graph. And then I'm going to be interested in unitigs. So these are non-branching paths of one of these de Bruijn graphs, highlighted here in red. As far as I know, there's no known practically efficient way to count the number of distinct unitigs as a function of k. Basically, you can, let me see, look at Ryan, see if he's nodding in agreement. Okay, cool. So basically, if you wanted to do this, to try to get at a problem of what's the right k should I use for my assembly problem, you might have to, well, just exhaustively run through them all. Compute a de Bruijn graph, look at the unitigs, compute the de Bruijn graph, count the unitigs, et cetera, et cetera. That's going to be rather annoying. And then similarly with overlap graphs here take, instead of k-mers, well, you can do it with k-mers, but usually you do it with reads and you look for tailhead overlaps of reads. So for example, if I take this genome and I pretend my reads are all the 7-mers, this would be an example of a overlap graph. And here again, in an overlap graph, which is the competing assembly technique to de Bruijn graphs, there's no known efficient way to look at large sort of geometric features of these graphs like vertex degrees. Okay, so in some areas, these k-mer-based objects are easy to describe as a function of k. Other ones, it's more difficult. So we need a data structure that's going to allow us to compute these sorts of quantities efficiently for all k size. And so that is our Prokrustean graph. So a TLDR of what the Prokrustean graph is, it's a graph. You build it from sequences, and you can use it to compute any of these k-mer-based quantities that I just talked about. Counts, frequencies, distances, et cetera, et cetera. And you can do this for all k simultaneously in time and space. Big O of the number of maximal repeats. So it lets you be able to do a lot of stuff in a somewhat compact representation. By the way, I didn't say this at the very beginning. If there are questions along the way, please do stop me. You don't have to just keep them for the end. Yeah?
I was just curious, how computationally intensive would this be for a data set which is large?
Yeah, so depending on what you're trying to do, which of these, it can be computationally extremely intensive. So for example, if you have a huge, deep metagenomic sequencing set and you try to form an overlap graph, you're going to have a very bad time. Other ones, like doing the counts and the frequencies, these are well studied, and we have very efficient techniques to do it. Yeah. So unfortunately, it depends. Okay, so For this talk, what K to pick? Let's just make it easy to check them all. Okay, so what is the Prokrustean graph? A couple of definitions first off. U is going to be my set of sequences. These could be reads, these could be genomes, something like that. So here, my running example, I'm going to have 3 sequences in my sequence set. Important definition number 1, maximal repeats. I have found that there's a lot of variation in how people define maximal repeats. So for this talk, it is going to be repeats, so it occurs more than once in your sequencing set, and any extension of that repeat has a lower count. So for example, this blue highlighted region right here, notice this AACAA shows up twice, so it's a repeat. And if I were to try to extend this one to the left, the count goes down from 2 to 1. If I try to extend this to the right, the count goes down from 2 to 1. Therefore, that blue highlighted region is a maximal repeat. Yeah?
Is it what people call math or something?
It's not quite a maximal exact match, mainly because instead of looking at matches, it's extension defined. Second important definition, locally maximal repeat regions, which is a mouthful. So it's talking about a region, so a region of one of these strings right here, and that has a corresponding substring S prime. So a locally maximal repeat region is a substring of some parent string whose count is higher, and any extension makes it have the same frequency as the parent string. Turns out that all these locally maximal repeat regions are a subset of these. So here again, this blue region right here is a locally maximal repeat region. This blue string, AACAA, shows up more than its parent string, but if you make an extension, it shows up just as equally. Okay, no more hard definitions after that. What is this Prokrustean graph? The vertices are easy to define. Take your sequencing set and take all the maximal repeats. And it turns out maximal repeats are easy to find in linear time. So here are the vertices of my Prokrustean graph. I have all my sequences, and I have my maximal repeats. Just for visual brevity, I have not included A, C, T, or G, those individual characters, just because it introduced a ton of arrows, as you'll see in just a minute. But imagine that they are there. The edges are going to denote this property right here between a string and its parent string. So for example, here I have this maximal repeat, and in this sequence, that region is a locally maximal repeat region, so I draw an edge from here to there. Same thing with this one over here, I draw an edge, and I keep on doing this. For each one of these regions, that's a locally maximal repeat region, I draw an edge. Okay. You don't have to keep the sequences around if you don't want to. And indeed, in our most efficient implementations, you just keep track of the start and end of the different regions and index the number of strings that you have with integers. But since we're looking at an example here, I'm going to leave all my sequences there. Okay, question. This tumor right here, what is its count? What's its frequency in this string set? Yeah, 1, exactly, it's 1. And you can figure that out just by saying, okay, I'll scan over and look for all my GAs. Okay, I only see one of them. But note, you could also figure that out by the fact that, hey, look, this first character right here is in a region that's not covered by blue. What does that mean? Oh, it's not part of a repeat region. Well, if it's not a repeat, that means it's unique. Only occurs once. Oh, and same thing with that 3-mer right there. It's not fully contained in the blue region, so its count is going to be 1. And in fact, all of these k-mers, okay, 2 to 5, are going to show up just as frequently as this parent string right here. So it gives us a handy way to group these similar k-mers across different k-mer sizes, which is quite nice because now we can sort of track the behavior of which 5-mers and 2-mers are sort of doing the same thing in my sequencing set as I change my k-mer size. One of the reasons why this data set is compact is because it disjointly covers all k-mers. What do I mean by that? Well, let's consider the k-mer AAG. Here again, this last character is not covered by a blue region. Its count is 1, it's unique. This is the only place AAG is going to show up. Let's consider another k-mer like CAA. Well, at this level of the Prokrustean graph, this CAA in each of these 3 occurrences is fully contained in a blue region. Therefore, it's a repeat. You know, it shows up more than once. But if you trace that location as you move down in your Prokrustean graph, we notice this k-mer is still completely contained in this blue region. And then again, as we keep on going down, aha, now this CAA is not fully contained in a blue region. It has the C character that's hanging off. So if you keep track of these regions that aren't fully contained in the blue regions, each k-mer in your sequencing set will show up once and only once in each of those locations for all k sizes. Let me pause there. Have I lost people? Okay. If we continue with this idea and look at multiple k sizes, we end up basically with these stacks of k-mers. So for instance, we looked at that tumor just a bit ago. Let's look at all the tumors that are not fully contained in a blue region. So for instance, this TT, this TA, this TG, that GA. All the tumors show up once in these red regions and only once. You do the same thing with the 3-mers. Look at all the 3-mers not fully contained in the blue region, and the 4-mers, etc. So you end up with these interesting looking stacks that turn out to have an extremely desirable property. Note, there's sort of 3 patterns of these stacks. This sort of stair step, where it's extending by 1. You have this one where it's sort of a pyramid. It extends both on the left and the right. And then this one where it's reaching both ends of the sequence. Those 3 classes of these sort of stacks of k-mers right here have a really cool property. We'll call them type 0, 1, and 2. 0 is where that stack reached over both ends. 1 is where it's a staircase. And then 2 where it's one of these pyramids. Look at this column K right here. K is going from the bottom to the top from 1 up to 9. Let's look at all the k-mers in this region and keep track of their counts. For this type 2, as I increase the k-mer size, the number of distinct k-mers goes up by 1. For the stair step 1, as I increase the k-mer size, the counts stay the same, 4 and 4. And then here, as I increase the k-mer size, my count goes down by 1. Interesting little thing is, back when I said the counts, the behavior is non-monotonic. Basically, we're taking that idea and applying it locally to each one of these regions that's not fully contained in the blue one. So, as a consequence of all this Prokrustean graph actually implicitly contains all compacted de Bruijn graphs. And you can kind of see this from this picture if you're looking at the 3-mer de Bruijn graph. That's just tracing along those particular edges. Okay, great. Don't look at this algorithm. To give you an idea, just a flavor, sort of fuzz your eyes out, and I'll draw your attention to just a few things. If I wanted to look at counting the number of distinct k-mers for all k-mer size, what do? I iterate over all these sort of stacks that I have, and I know the behavior of the function of k. If it's type 1, type 0, or type 2, there's change of +1, 0, or -1. So just go through it and keep track of all those changes. And at the very end, apply all those changes, and you're done. So you can see how you could get all k-mer counts for all k-mer sizes with one pass over this entire graph. Try to do this with other sort of modern indexes, like an LCP or FM-based index, leads to a lot of inefficient jumping around. Whereas with this, you just need one pass over this graph. Okay, that's cool and all nifty and all. Questions before I talk about the construction? Okay. Vertices, as I said, are easy. These maximal repeats have been known how to compute efficiently for quite a while. The edges are a little bit more tricky. In our publication, we give 3 different ways of increasing complexity and efficiency of how to construct this thing. I'll start with the easiest here to describe it. Basically, if you take an affix tree, what is an affix tree? That's a suffix tree. Then you take your string set and you reverse it and make a suffix tree of that. If you keep track of edges in your suffix tree or the reverse suffix tree, these correspond to blue regions that are contained in the left or the right of the sequence. And if you have an edge that shows up in both of those trees, that's something in the middle. So by looking and finding all these blue regions, that then allows us to figure out where to draw our edges in the Prokrustean graph. Okay. So let's talk about applications real quick. This is all fine and fun in theory. Does it actually help us do anything practically? Well, let's look at one of these classical things like counting k-mers. So let's take real metagenomic data sets and look at one of the fastest single k-mer counters out there, KMC 3.0 to be exact. So on short read and long read data sets, I want to look over an entire range of k-mer sizes. With KMC, you have no other choice but to run it at 20 and then 21 and then 22, etc. You sum up the time that took, it takes KMC about 105 minutes. Use this Prokrustean graph, since we're only passing over the graph once, it takes 30 seconds to do the computation after you invest 30 minutes in building this data structure. Similarly, if you try to do this huge range of k-mer sizes for this long metagenomic set, it would take days with KMC3, whereas a couple minutes after you invest just over an hour to index this thing. And the nifty thing is, this allows you to start looking at real data over a range of k-mers a lot more practically, efficiently. So for example, consider this one study that looked at infant fecal microbiome samples. They took a single k-mer size, and they had a bunch of different samples at different time points. And they wanted to compare the sort of the trajectory of these over time, so they looked at pairwise Bray-Curtis similarity between the k-mer counts. And they based their paper, off of those conclusions. However, if you look at the entire range of k-mer sizes, some really interesting things happen. Contradictory conclusions. Here, at a small k-mer size, samples 2 and 4, that is this green line, are the most similar, have the highest similarity. Or if you increase your k size, like way out here, 2 and 4 are the least similar. And so, you gotta be careful about which k-mer size you pick, because your conclusion might flip completely depending on what it is. So thankfully, computing these graphs can now be done quite efficiently with this Prokrustean graph. Okay, what about counting unitigs? So it turns out that in just a few minutes, you can get plots that look like this. As a function of k-mer size, how are the number of unitigs in your compacted de Bruijn graph? And since this is just stuff that we've done recently, I have no idea, but I have a strong suspicion that using these things might inform k sizes for different assemblies, just because we have some really interesting little dips in here. Okay, if you were to try to do this using an extremely fast construction technique for compacted de Bruijn graphs, it would take you a couple hours. Use a Prokrustean graph, making this plot takes you 28 seconds after you invest 30 minutes to build it. You may be familiar with variable order de Bruijn graphs, especially if you're in the assembly realm. So a quick little bit comparing and contrasting these two. Variable order de Bruijn graphs, at a very 10,000 foot view, basically take all your de Bruijn graphs and sort of just link them together. So you have these sort of shorter, longer operations to move between the different de Bruijn graphs. Using a Prokrustean graph, you don't need that. So you don't need these kinds of edges, and it's practically fast. So currently there are just a couple variable order de Bruijn graph practical implementations, one of which is this VO de Bruijn graph, which uses a certain data structure. And on a single human chromosome, it takes about 200 minutes and a gig of RAM to be able to construct this. Whereas if you use a Prokrustean graph on the complete chromosome 14 of a human, it's less than 8 minutes and about 100 meg of RAM. So the hope is that practically we will be able to use this Prokrustean graph to get much more efficient variable order assembly techniques. Okay, but not just boring graphs. This Prokrustean graph also has overlap graphs. So consider this sequencing set. Here is the overlap graph. The numbers on the edges tell you how much the tail overlaps with the head of the sequences. Wow. Yes. If you're interested in some sort of structural property of this graph here, you might be looking at in degrees and out degrees of these. These overlap graphs are classically known to be very annoying to try to compute because the brute force way to do it is quadratic in the number of sequences you have. But on a real metagenomic data set with about 4.5 billion base pairs, you can construct your Prokrustean graph in 30 minutes, and then just with a single pass that takes 1 minute over your graph, you can compute all of your vertex counts and your degrees. So you can get some idea of the structure of this overlap graph. Cool. So what we want to use this stuff for is improve assembly. So Spades, for example, uses 3 different k-mer sizes. And I've asked Anton about this, and he goes, they seem to work in practice. So it'd be nice to optimize and maybe formalize this to be able to use a whole range of different k-mer sizes if appropriate. And so build on previous work that tries to use variable ordered Boring graphs to do this. And the sort of vague idea that I have is maybe adjusting your k-mer size based off of local sequence content might be the way to do it. And thankfully, with this graph, you can do that on the fly. And then PSA for everybody. If you're using k-mer-based methods, don't ever use just a single k without poking around and looking at those other k's. You don't want to run into this situation where another analysis of your data says, hey, your conclusion is only because you picked that k-mer size, things fall apart in a different one. So it's important to look at the different perspectives. Cool. So to conclude, this is a data structure that makes k-mer queries efficient and convenient for multiple k-mer sizes. I hope it's going to unlock new assembly techniques. Very efficient in practice. It's implemented in C++, freely available if you want it. This is really the brainchild of Adam Park, my very talented master's student who is joining UCLA in a PhD program later this year. And then thanks also to the rest of my lab who also contributed to this work, especially in its applications. And thank you to funding agencies. I'll take any questions.